Hello, everyone. Welcome to this first climate seminar of 2024. It's a real pleasure for us to be with you, to start the year with you today with the continuation of our seminar series. For those who don't know me, I'm Charmilino. I'm project manager at CLIMACT, and I'm going to be today's moderator of this session. This is a special seminar today because it is dedicated to our 2022 CLIMACT starting grants that have been created to foster novel collaboration on research efforts toward climate change adaptation and mitigation. It's been roughly a year that the PIs have been working on their project, their starting grants, and today they will present how it went, what are the results so far, what are the next steps for them, maybe in the continuum of research they're working on. We were supposed to have 3 speakers today. Unfortunately, Horst Pick will not join us due to last-minute circumstances. Also, the 2 presentations will be in English. Don't hesitate if you have questions in French. I'm going to be happy to translate them if need be. Finally, to put a bit of context also in this presentation, both speakers today are from EPFL, Rizlan, Bernier-Latmani, and Gabriele, Manoli. But as part of the CLIMACT starting grants, to have a full interdisciplinary and interinstitutional level in the projects, we have also PIs from the University of Lausanne. So we say hello to Christophe Randin and Nadav Pelek. So before starting, I'm going to shortly present Rizlan with a short bio. Rizlan Bernier-Latmani is a geomicrobiologist with interests in geochemical and microbial processes, particularly microbial-mediated transformations in subsurface environments. She studied in the United States and came to Switzerland as a tenure-track assistant professor in 2005. She is now a full professor in the Environmental Engineering Institute at EPFL. Her work spans molecular scale mechanistic understanding to the interrogation of field scale processes. I'm going to stop sharing my screen, and the floor is yours, Rizlan.
Very good. So first, I'd like to thank everyone who's here for being here to listen to my talk, and then Gabriele is later. So this is, as Charmili said, a project that was funded through a CLIMACT starting grant. So I'm one of the PIs, but very importantly, the other PI is Christophe Randin, who's also here today. And the person who did the work is Aida El-Sangheri. She is a master's student at UNIL who came to my lab and worked on this project. So you see here a meadow, irrigated grassland in the Valais. And this is what we're going to talk about, irrigation practices and their impact on greenhouse gas emissions. Okay, so greenhouse gases, they don't need any introduction. We all know what they are: CO2, methane. Perhaps less well known is nitrous oxide, N2O. That's also, its concentration has also been rising since the Industrial Revolution. But contrary to CO2 and methane, it's not really clear why there's been this trend here, particularly in the past 10 years. What is fueling this increase in N2O in the atmosphere? There are several sources: agriculture, industry, combustion, and wastewater treatment. Importantly, N2O is 265 times more potent than CO2. So while this concentration is much lower, thankfully, it's much more, it has much more of a warming effect than CO2. So I've highlighted or italicized agriculture and wastewater treatment here because both of these are microbial sources of N2O, and that's what we're going to focus on today in the context of soils. So if you allow me, I'm just gonna delve a little bit into microbiology to provide the context for what we're looking for here. So biological transformations of nitrogen, for example, in soil, generate nitrous oxide via 2 major processes. One is denitrification, and I've depicted here a microorganism, this red thing here with a little flagellum. The organisms have proteins, these little colored squares along the outside of the microorganism. It's actually inside the membrane, but it doesn't matter. And so what happens in denitrification is, in the absence of oxygen, nitrate, which is this compound here, is being transformed, reduced for those who are in the know, reduce to nitrite, to nitric oxide, to nitrous oxide, and ultimately to N2, to dinitrogen gas. So this process, as you see, is stepwise, and each one of the steps is catalyzed or accelerated by a given enzyme, protein, and these are labeled, the corresponding genes are labeled here. So NER, NOR, and NOS. So just not super important, but just at the end, I will come back to these genes because they allow us to then monitor the activity of microorganisms. In any case, the important thing to know about denitrification is that it happens in the absence of oxygen, and that N2O and nitrous oxide is not the end product, but essentially diffuses away from the cells in the process of this reduction, stepwise reduction. On the other side, we have nitrification, the second major process. And this, in contrast to denitrification, takes place in the presence of oxygen, not only in the presence, but it requires oxygen. And now the substrate, instead of nitrate, is ammonia. And it's being, instead of reduced, oxidized. For those in the know, this is an electron donor. And so it produces hydroxylamine. And hydroxylamine can either chemically disproportionate into N2, into nitrous oxide, or the next step, which is the production of nitrite, can also yield N2O. So essentially, these two processes, and then the same as before, the protein here is AMOA and the corresponding gene is here. So essentially, these processes are very distinct because one of them, at the high level, we can say denitrification happens in the absence of oxygen, nitrification requires oxygen. And so there are two different ways, different regimes in which, through which N2O is produced. So now let's talk about soils, which is what we're interested in. So for denitrification, well, we said it can't have oxygen, so it happens mostly in, so this is my little depiction of a soil. These are the particles, and the space in between the particles is filled with water in blue. Okay, so this is a saturated soil. Obviously, if it rains heavily or there's a lot of water that's added to the soil, you will have saturation. And hence, if you zoom in here, these little microorganisms will not have oxygen, right? And so their nitrate can be reduced to N2O. So this now we've sort of linked the microbial process with the hydrological state of the soil, right? So this is when there's a lot of water. And in contrast, for nitrification, you see the same type of diagram, little particles. In between, you have water in blue, but you also have air, and that's in white or no color. And so when you have a soil that is not fully saturated, that's wet, but not full of water, as a result of moderate rain or moderate irrigation, then there is a possibility of having nitrification where ammonia is then oxidized to N2O, to nitrous oxide. Okay, so under heavy rain, you have one regime and the other one is the other. Now, the thing is that it's known that when the heavy precipitation falls on a dry soil or irrigation water is added to a dry soil, we have this massive production of N2O, so-called the hot moment. Also, it's true also for carbon, for methane and CO2, etc. It's so-called Birch effect. And so essentially, the paradigm in the field is that if you have steady, non-saturated water content, then you minimize N2O production. Whereas if you have a rapid and large variation in soil water, you have these hot moments where you have massive production of N2O. What we don't know is in these hot moments, whether it's this production of N2O is due to denitrification, nitrification, or both. And this is, being debated. In some cases, people say it's denitrification, others it's nitrification. Likely, there's some combination of the two depending on the specific conditions. So essentially, Christophe and I were talking, as one of these CLIMACT events. We met through CLIMACT, by the way. And so, we were thinking, okay, so he works in these irrigated grasslands, and he works on how much irrigation water is delivered. And I work, on various things, including N2O emissions. And so we thought, well, maybe what we need to do is team up and try to figure out how what he's doing anyway, which is controlling irrigation in grasslands, will impact greenhouse N2O emissions. So Christophe and others have put together this project called ODEAL, which essentially optimizes agricultural irrigation in the Entremont region in the Valais. And these are particularly, this is focused on irrigated mountain grasslands, which, you know, generate all this wonderful cheese, like raclette cheese from the region. And what they were interested in is to minimize water use by the farmers, because, you know, what the farmers do, they just open the sprinklers and then they let them go for a couple of days, come back and turn them off. And so there's massive amounts of water that are used to irrigate these grasslands. And with the ODIL project, what they've shown is that if you actually monitor the moisture content, and that you only irrigate when it falls below the permanent wilting point, which is a certain level of water content. This is the water level below which plants won't be able to obtain water from soil. So if you keep this water, soil water moisture at a level above this permanent wilting point, but not too high, and you only irrigate when needed, essentially, then you actually don't decrease the yield of grass. And of course, you save a lot of water. So this is this ODIL project. And so, you know, when we discussed this, I was like, well, this is perfect. This is perfect for us, the microbiologists, to piggyback on, to look at whether when you change this irrigation regime, whether it also impacts N2O emissions. And a hypothesis was that if you have more steady water content over time rather than up and down, you will have less N2O emissions. And so the ODIL project is, you know, uses automated water sprinklers that can be turned on remotely when this water content, soil moisture content drops below a certain level. So with this, we thought, okay, perfect. Let's set up a field site where we have 3 treatments. So this is you can see a picture of the field site. Treatment A, where we have no irrigation, so no added water, only precipitation. Treatment B, where we control the irrigation, a la ODIL. And treatment C, where we have the irrigation like the farmer would normally. So this is an actual irrigated grassland with a farmer who uses it. So he allowed us very kindly to do this experiment on his field. And what the objectives were to measure N2 gas emissions, so the N2O flux, and to characterize microbial activity in the soil with the very simple but complex to answer question: does controlled irrigation result in reduced N2O emissions? So the way to measure N2O gas emissions are using these types of chambers. So you put the chamber on top of the soil and measure how much N2O accumulates in the chamber. So if you put the chamber on for an hour, every 15 minutes, take a measurement of the N2O concentration, and then you get a straight line. And using the information from this, we can calculate the flux, which are N2O, moles of N2O emitted per surface area per unit of time, so nanomoles per meter squared per second. And so just to give you a visual of this, so we have our soil moisture, we have our rain gauge in the field, and then here we see, I think it's Aida, this is the chamber, so this is quite low tech. We don't need really high tech to do these measurements. And so she's collecting here gas phase from the chamber, and then she's transferring into a vial that is gas tight, so then we can measure this on the mass spec later. Okay, so do this every 15 minutes for each chamber for a duration of an hour over a number of months. Okay, so these are the results. Precipitation, but meaning rain and irrigation. So in the case of this is treatment A, where there's no rain, no irrigation, so only rain. And in these bars, you have the rain, so precipitation, and the amount in millimeters. And in orange, we have the N2O flux, so how many in nanomoles per meter squared per second, okay? And we calculated that this particular treatment received 175, almost 176 millimeters from rain, and emitted 6.3 milligrams of N2O per meter squared. Now the second treatment, so this is now our controlled irrigation with ODIL. So again, it's the sum of total water that's added, it's irrigation plus rain. And we see that there's more water, that's 322 millimeters amended over the period from July to October. And the amount of N2O emitted, it has increased to 7.9 milligrams N2O per meter squared. And then finally, we have the high irrigation. So this is the one, the farmer, basically mimicking the farmer's practices. And we see there's much more water, 727.6, and much more N2O, or this should be a per meter squared, much more N2O emitted. So, so 1.8 times less N2O is emitted by the controlled condition versus the normal irrigation, normal meaning farmer practices, and much less water, 2.25 less water is added to the controlled irrigation versus the high irrigation. So it suggests that we go in the right direction if we control the amount of water that's amended to these irrigated pastures or the irrigated grasslands, then we decrease the amount of N2O. And so, but it's not a lot, right? 1.8x, we're expecting to have a much more impact. And so then comes the reality of running a field experiment. So this is soil water content as a function of time. And you can see that for the no irrigation control, there's quite a huge change in soil water content, and it goes down to below this threshold, this wilting point, permanent wilting point, see several times. In contrast, in blue here, we have the high irrigation, so the farmer irrigation, and we see that this is increasing, especially at the end here, there's more water, and it's getting to field capacity, which is defined as the amount of water retained by the soil just for free drainage. And then in between, in green, we have the controlled irrigation. And what we see is that actually, unfortunately, we had hoped to target about 85 or so as the millimeters, as the point where we would oscillate around this content, this water content as a function of time. But unfortunately, due to a number of reasons, this oscillation was much larger, and it resembled the high irrigation, unfortunately. So the difference between the soil water moisture content between the high and the controlled irrigation is not as high, as we'd hoped. And the target here was this steady. We wanted to basically control the water content to a more or less stable value, which we would think produces the least amount of N2O. And so there's a number of technical difficulties. I'm happy to talk about this, if they have questions, but this is why we couldn't have as good control. But this is something that's completely doable. It's just that, given the situation, we weren't able to do better. But still, we were able to see this decrease in N2O emissions. And so then what's ongoing now is the microbial characterization. Remember, I showed you this sort of portrayal of the microorganisms and all the genes that are involved in either denitrification or nitrification. So we took these soil samples in August and in September, which correspond to approximately sort of a high flux and low flux for all cases, and we're hoping to then look at how much denitrification and nitrification microbial activity there is, and this is ongoing. So what we were doing, this is a core that we collected. We sampled the DNA, the soil, we preserve it so that we can extract RNA, and the RNA provide information about the activity of the microbial community at the time of sampling. And then we'll quantify the abundance of the genes that I mentioned before that are markers for these two different metabolisms. So with this, I'll come to the conclusion, which is, can we answer the question that we set out to ask? So so far what we can say is that the data we collected suggests that indeed controlled irrigation, that is to say, using less water and keeping the moisture content as stable as possible, has a beneficial impact on N2O emission, that is to say, decreases them. And then the other question was, do we have more denitrification or nitrification? We can't answer that question yet, since we're in the process of obtaining these data with QRT-PCR. So with this, I'd like to thank you for your attention, and I'm happy to answer questions if there are any.
Gabriele Manoli is a tenure-track assistant professor from ENAC at EPFL, where he leads the Laboratory of Urban and Environmental Systems. He received his PhD from the University of Padova in Italy in 2004. In 2000, 14, I'm sorry. And before joining EPFL, he was a postdoctoral researcher at Duke University and ETH Zurich, as well as a lecturer at University College London. Gabriele, the floor is yours.
Thank you very much. Let me share my screen. Okay, good morning, everyone. Thank you very much for the introduction. Thanks also, Rizlan, for the nice presentation. I will now talk actually about the ego-decovariance tower, so precisely what you would like to do next. So yeah, today I have the pleasure of presenting the results of this CLIMACT project titled Monitoring Urban Ecosystem Services, which was led by Professor Nadav Peleg from UNIL. Actually, Nadav was supposed to present today, but unfortunately, he couldn't be here, so I'm presenting the project. But for both of us, of course. So, as you know, most of the world's population now lives in cities, which are projected to continue growing in terms of population, in terms of activities. And so the combined effect of urbanization, of course, global climate change, which is acting more at the global level, bring to the forefront the need to plan and design more sustainable cities, more sustainable and livable cities. So in a sense, I think we are all familiar nowadays with concepts like green cities, sponge cities, carbon neutral cities. These are very hot topics that we hear every day now. And really the idea is to, of all these new urban planning frameworks, in a sense, is to use nature-based solutions in order to address some of the pressing problems that we have in cities, from mitigating extreme heat during the summer, to increase urban water storage and reduce the flood risk, to reduce also carbon emissions, CO2 emissions. So, however, you know, all these efforts are typically guided by aesthetic and qualitative principles often. And in a sense, what is missing is a quantitative understanding of all the underlying processes, physical and anthropogenic processes that regulate such dynamics in complex systems. So what, you know, it's crucial is really to quantify and understand water, carbon, and the energy budget of cities, which are very complex systems, you know, where you have a complex 3D structure of the urban fabric, you have anthropogenic activities, and so on and so forth. So just for example, like to give you an example of how the different fluxes and variables that are involved, we can look at this figure here on the right, where you see Lausanne. And if you think about the energy budget, cities compared to natural environments clearly modify how radiation is reflected, is emitted, how it is partitioned between sensible and latent fluxes, which then regulate the microclimate of the city. So typically during summertime, cities are much warmer because the built environment, the absence of vegetation, cause higher sensible heat fluxes and higher air temperatures and surface temperatures. From the water side, you have precipitation, and the sealing of urban surfaces reduces the infiltration, so you have much higher surface runoffs. And of course, introducing vegetation in cities can ameliorate this, can allow more infiltration and reduce runoff and the risk of flooding. From the side of carbon, you of course know that, you know, all the anthropogenic activities from traffic, buildings, and so on and so forth that we have in cities are generating carbon emissions. And in a sense, vegetation can act as a carbon sink. So these are all the potential benefits of vegetation in a city. How can we quantify this? One of the most reliable ways to collect direct measurements of the urban energy, water, and carbon budget is, again, as Rizlan mentioned, ecovariance towers. Because ecovariance towers, such as the one illustrated on the left here, allow to collect sub-daily observations of radiation, energy fluxes, so sensible and latent heat fluxes, as I mentioned. You measure temperature, humidity, wind speed, CO2 concentration, and so on and so forth. So you really get an overall picture of the key variables and fluxes that regulate microclimate and hydroclimate of the system. So the goal of this project was to set up a sort of open-air laboratory in Lausanne to monitor all these variables via an ecovariance tower, really with the long-term idea of using such information in combination with models and other work that both me and Nadav is doing to inform planning and decision-making in terms of introducing and using nature-based solutions in urban environments. However, unfortunately, the original plan had some, we had some issues. So the original plan was to deploy the tower on the UNIL and EPFL campus as a test bed. And then because of the portability of the sensor that we purchased, the idea was to move it to different locations in order really to get information from different types of urban settings. Because as you can imagine, central Lausanne is very different from the EPFL campus, which is very different from the suburban and rural areas. However, due to delays in the supply chain, some technical issues, so we received the sensors very late. So, I'm afraid my presentation today, I won't have much results to present because we really had issues with the setting up of this. So we mostly present, the state at which we are with an outlook of what we plan to do, which unfortunately we haven't been able to do within the year, but we plan to do very soon in the coming months. And in fact, at the moment, we're basically, at the stage of calibrating the instrument. We now start having some master students who are going to work on this. So let me show you some pictures, first of all, of what we did. So here is the Geopolis building, and we installed this flux tower when it arrived, again, very late, unfortunately. So we installed this flux tower on the rooftop of the Geopolis building. And the idea was to use this as a first test bed. And here you see an example on the left of raw data that we are collecting. In a sense, I know that the legend, so you don't really see the numbers, and that's done on purpose because since we are not really calibrated the instrument yet, we do not trust the numbers too much. So I wanted just to show you an example of time series of air temperature, CO2 concentration that we are currently collecting, but again, that we are in the process of analyzing and calibrating properly. But on the right, you see an example of what our plan is really to get long-term time series of, as I mentioned, all these different variables, so from temperature to CO2. But on the right, you see this example of latent heat fluxes measured in Singapore and Melbourne using a very similar setup by colleagues, and we plan to do the same here in Lausanne. And I'm showing the latent heat flux because, as I mentioned, is a key variable to understand how vegetation, the role of vegetation in regulating the microclimate through evaporative cooling and so on and so forth. And here, for example, you see the diurnal cycle, the long term, so the annual variability of latent heat fluxes, and the comparison between observed and simulated, because the idea is really to use this observation in conjunction with the urban ecohydrological model that we recently developed with colleagues from ETH and Singapore, and use observations to constrain the model and run scenarios and better understanding the overall energy budget of the system and how different urban characteristics contribute to that. Another example of possible application, this type of measurements, so EC covariance observations, can also be used to improve high resolution estimates of CO2 fluxes from buildings, traffic, vegetation, and so on and so forth, as I mentioned. And on the left, you see a recent study from the city of Basel. And the map, the red colors, basically show the estimated CO2 fluxes from different part of the city, and you clearly identify buildings and roads where you have higher emissions, but you have also have the carbon sink in green from the vegetation. And if you look at the image, I don't know if you see my mouse, but the blue contour line basically shows the footprint of the eddy covariance tower. So an important aspect, especially in urban environments, which are highly heterogeneous, is to properly understand what is the footprint of the flux tower. So what are you measuring? So where the information that you are collecting are coming from. And this is precisely what we are doing at the moment. So our site is on the right, as you see. So that's the Geopolis building. And we are in the process now with the master student to calculate the footprint. And ideally, the next step is to do a very similar assessment. So have a high resolution assessment of the emissions from the very interesting setting that we have here, where we have the campus, the UNIL campus, but you also have vegetation, we have traffic from highways, we have construction sites. So there is a number of potential sources and sinks of carbon that should be properly quantified. Another example of application that for which we are using this data, and we plan to use in the near future, as soon as the instruments are properly calibrated, is this study led by Aldo Brandi, who's a postdoc in my group. So we are basically studying the potential for wind energy in cities. And this requires to simulate wind speed and, of course, all the complexity of urban climate in urban context. And of course, the flux tower will be instrumental to compare not just with wind speed, but having a ground confirmation for all the different energy fluxes that we simulate with the model that we want to calibrate and validate in order to trust the simulations. As I mentioned at the beginning, so we are starting from the UNIL and EPFL campus as a first test bed, but really the plan, because of the portability of the tool, is to then have an instrument that we can move to different sites. And for example, I'm involved in the PAPUS project, which is a Synergia grant led by BSL that is exploring biodiversity and nature contributions to people in cities. And the project has a number of study sites in Zurich, Geneva, and Lugano. So really, this is the perfect candidate, in a sense, for the future deployment of our flux towers. So as soon as we learn and we are confident with the work that we are doing here on campus, we will move it to different locations, for example, within the frame of this project, precisely to again assess how urban vegetation and different combination and assemblages of urban vegetation are actually influencing the water, carbon, and energy fluxes within the complexity of the urban fabric. Last but not the least, Professor Peleg at UNIL is also carrying out a number of boundary layer measurements using radiosones, as illustrated here. So you see the vertical profile of temperature, humidity, and wind speed. And again, the flux tower here is necessary, and we plan to use it in combination with this type of measurements because we need to constrain the surface conditions in order to understand what is then driving changes in the profiles, in the vertical profiles of boundary layer characteristics. So thank you for your attention. This was a quick overview of the project. I'm sorry we couldn't present any concrete result. Unfortunately, mostly an outlook, but I'm now more than happy to answer any questions you might have, and of course, I'm also happy to explore collaborations. Thank you very much.